Learning objective 2 is to describe the physical structural support of the central nervous system. So what are the structural layers that help to make up the brain and spinal cord? So the physical support of the central nervous system is one, the skull. Okay, I think the skull, if I remember this correctly, is one of the first units you did in anatomy lab. You talked about the bones of the skull, the frontal, occipital, parietal. All of those bones help to form the cranium, right, which houses the brain. So that hard, thick nature of the skull protects the brain. The brain is very delicate. Hopefully you were able to go into the lab and touch it. It's a very delicate tissue, and so we want to have a really solid case or covering to protect the brain. Same for the spinal cord. So the spinal cord is protected in the vertebral column, right, which is a series of vertebrae that is stacked on each other to help protect the spinal cord all the way through the length of the spine. And then within the skull, so deep to the skull, we've got the meninges. And the meninges are divided into 3 different layers. We've got the dura mater, which is Latin for tough mother. As you would imagine, this is the thicker, tougher, outer, more protective layer of the meninges. Deep to that, we have the arachnoid mater, which is Latin for spider. And that's exactly the way the arachnoid mater looks. It's more of a meshwork, of a network, and not necessarily a thick or continuous or tough layer as we see with the dura mater. In the arachnoid is where we have things like the blood vessels, and then deep to the arachnoid, but above the pia is where we have that cerebrospinal fluid. So in between the arachnoid mater and the pia mater, right, in this space here, this is called a subarachnoid space. And this is where we have the cerebrospinal fluid, right? All throughout the brain, all throughout the spinal cord, in between these 2 layers is that thin layer of cerebrospinal fluid, which helps to coat and protect the spinal cord and brain. And then the deepest layer, which is the most intimately connected to the spinal cord and brain itself, is called the pia mater, also standing for or translating to tender mother. So in the outer layer, we've got the tough mother, more fibrous, more thick connective tissue. And then in the pia mater is a very thin, very delicate layer that's really inseparable. It's one with the brain and spinal cord tissue. So here's what some of those structures look like. So here is the scalp. Remember, the scalp is going to be the skin over the skull. Then we have the skull itself, and that can be further divided into all the different bones that you would have learned about. Deep to that is the dura mater, right? That thicker, tough outer layer. The dura mater also has certain modifications where it can divide the 2 cerebral hemispheres or divide the cerebral hemispheres from the cerebellum below. So the dura mater will have certain folds and projections like the falx or the tentorium that help to divide certain parts of the brain. Deep to that is the arachnoid mater, which we said is more of a meshwork. And then deep to that is the pia mater, which is really intimately connected to the brain. Okay, now notice all of these extend all the way through the length, even down to the spinal cord. So the same 3 layers will be present as we go from superficial to deep at the level of the spinal cord, all the way through to the sacrum. Okay, here's another view. Here's a cross section of the spinal cord. So here are the parts of the vertebra, the vertebral body, the vertebral arch back here. Here is the spinal cord itself. So here's the cross section level that we're looking at. Here are some of the spinal nerves that are exiting left and right. And then here's the actual spinal cord. So we will speak about the difference with gray matter and white matter. This is one of the interesting things about the brain versus the spinal cord. At the spinal cord level, gray matter is going to be internal. White matter is going to be external. And white matter simply means that there's more myelin, right? That's why it appears white. In the brain, as you'll see here, the gray matter is actually over the top. It's more superficial. The white matter is actually going to be deeper. So this is an interesting contrast that we see with the spinal cord versus the brain, where in the brain, the gray matter covers over the white matter. In the spinal cord, they reverse, and the gray matter is inside and white matter is on the outside. Also, I want to point out on this slide the presence of the arachnoid villus. The arachnoid villus are these projections of the dural sinuses that help to reabsorb cerebrospinal fluid. So cerebrospinal fluid is being made by the ependymal cells that line the ventricular cavities, and then it's going to circulate in that layer that we talked about, and then it's going to need to be reabsorbed to be recirculated. And so the way that it's reabsorbed is via the arachnoid villus. Additional physical support for the central nervous system is the CSF fluid, which is in the ventricles. It's a clear, watery fluid that helps to bathe the brain and spinal cord. It's made by the ependymal cell, so one of the 5 glial cells that we talked about earlier. And the ependymal cells will congregate themselves to form what's called the choroid plexus. And the choroid plexus is a structure that's found within the ventricles that helps to make that cerebrospinal fluid. It's going to release that into the subarachnoid space. So we pointed out where that was earlier, and then it's going to circulate throughout the ventricular system and then become, and then be reabsorbed, by the arachnoid villi. The function of cerebrospinal fluid is to cushion the brain. It's going to help to prevent the brain from sloshing around. So while we talked about the skull being a nice, hard, protective casing, the skull is dangerous. It can actually injure the soft brain tissue itself. So having that fluid helps to just suspend the brain in that fluid and sort of cushion the movements, especially when we move the head, but also just prevent it from sloshing or hitting against that skull. Another thing that the CSF does is maintain a stable interstitial fluid environment, right? So the fact that it can be made and then be absorbed allows us to keep a steady state as far as the amount of glucose, the amount of electrolytes, sodium, potassium that exists within that fluid. All right, so on this slide, here's an example of what cerebrospinal fluid looks like as far as the comparison to the makeup of plasma. So here's plasma, which is the watery component of our blood, and then here is CSF. The trend that I want you to take away from this, so I don't want you to memorize this necessarily, but take away the trend that glucose and proteins are at much lower levels in the cerebrospinal fluid than they are in the plasma. So much lower levels of glucose and much lower levels of proteins, especially proteins, right? Especially proteins, a very significant deficit when we look at plasma compared to CSF. And that is because of the blood-brain barrier. So the blood-brain barrier, which is formed by the ependymal cell, or the astrocyte, is what helps to eliminate or prevent certain large bulky molecules like glucose and like proteins and even erythrocytes from escaping into the interstitial fluid and into the nervous tissue. So these are the 2 things that I really want you to pay attention for. The others are relatively the same. So relatively the same amount of sodium, potassium, chloride, and then calcium in both of these fluids. All right, I'm not going to go into too much depth with the ventricular system. Again, I suspect you did this in lab. So as far as just pointing out where these ventricles are, the lateral ventricles are the 2 largest ventricles. The 3rd ventricle is going to be around the thalamus here, sort of around the midbrain region. And then the 4th ventricle, closer to the pons, is going to be the smallest of the 4. So this is the route that CSF takes. It's made in the choroid plexus of the lateral ventricles, then it goes to the 3rd ventricle, then down to the 4th ventricle, then through to the spinal cord, as well as around the brain, and then it gets reabsorbed by the arachnoid villi, which are going to be at the top of the brain. The sinuses at the top of the brain. Okay. Let's move to the 3rd learning objective to describe the nature and significance of the blood-brain barrier. So we've said a little bit about the blood-brain barrier. Let's talk about the actual structures that make that barrier up. So the central nervous system is about 2% of our total body weight, okay, which is really quite interesting. The brain is about 3 to 4 pounds in weight. But it gets so much of our cardiac output. It gets so much of the blood supply that's released from the heart. And so we can think about the brain as being a vital organ in that way. It's relatively a small percentage of our total body weight, but it gets a high relative percentage of the total amount of blood supply from the heart. And the reason is because of the high metabolic rate. So the brain uses about 20% of the oxygen that is consumed by our body at rest, and about 50% of the glucose that is consumed by our body at rest as well. Okay, so let that sink in for a minute. Okay, the brain being about 2% of our total body composition takes 50% of our energy. Really amazing, right? To think about how important our brain is, but also how much of a energy requirement it has because of the main role it plays in our general body functions, even at rest. Even while we're resting, the brain has to send instructions to the heart, to the lungs, to the kidneys, the liver, to do all of that processing that is outside of even physical movement or physical behavior. So it's going to depend on a lot of blood flow, mainly for its high demand in energy. Okay, blood supply to the nervous system is going to depend on aerobic glycolysis. We talked about the difference between aerobic and anaerobic glycolysis back in cellular respiration, but it is the source of ATP that relies on oxygen, right? Remember the electron transport chain and the oxidative phosphorylation. This is what the brain really needs. It's dependent on oxygen-derived ATP production. And so it's going to take up a lot of our glucose, a lot of our oxygen. It doesn't have the ability to store glycogen, but it does have the ability to make glycogen, specifically by the glial cells that we mentioned, like the astrocytes, which have the ability to make some glycogen, but not store it for long-term use. Fatty acids cannot be used for energy in the brain. Ketones can be used, but only during extreme conditions. So if you think about going for long periods without food and going into somewhat of that starvation mode or ketosis mode, then very rarely your brain can tap into those stores, convert those ketones to glucose, and then use that glucose for energy. But under typical conditions, your brain really prefers to have glucose. If we think about the loss of blood supply, this brings up strokes. So strokes are whenever we have a decrease in blood supply to the brain. There can be two different types of strokes. An occlusive stroke is where we have a blockage of a blood vessel, okay? Typically seen with things like atherosclerosis from smoking or poor lifestyle choices. And so the arteries and the capillaries of the brain narrow to the point of occlusion. So that's going to be an occlusive stroke. A hemorrhagic stroke, on the other hand, is going to be more associated with a rupture of a blood vessel. So think about things like extreme chronic hypertension, which is where you have high sustained blood pressures that really weaken the walls of the vessel and result in rupture of the vessels. And so you're going to have a bleed in the brain. So both of these types of strokes will obviously have some of the same types of consequences. Loss of memory in some instances, loss of immediate physical function, loss of cognitive function. Patients will typically notice that they can't move their face, can't move one side of the body. Depending on what side of the body we have the injury on, the opposite side of the brain would be affected. And so these are some of the manifestations that we see if we lose blood supply to the brain. The blood-brain barrier, this is formed by the capillaries that go to the brain. It's going to be a site of exchange for important materials from the blood and the interstitial fluid. It's going to be made by a single layer of endothelial cells and then guarded or separated by tight junctions. We have talked about tight junctions before. Remember, tight junctions help to prevent any leaking of material in between the sides of the cells. Any type of exchange needs to happen through the apical surface and then the basolateral surface and then into that, that blood vessel or that interstitial fluid. So tight junctions really help to secure the communication in between the sides of the cells And the blood-brain barrier is a special anatomical junction that helps to limit that exchange. So it's an additional guard, an additional barrier on top of the tight junctions that further limits what can move back and forth in between the interstitial fluid in the brain and the blood vessels that go to the brain. And as we mentioned earlier, it's formed mainly by these astrocytes. Okay, so here's two different examples. One is a typical capillary. So this is going to be anywhere else in your body. Think about your heart, your lungs, your muscles. This is what the endothelial blood vessels will look like. And then here are what the blood vessels look like specifically in the brain. So notice we've got the blood vessel here. Okay, we've got tight junctions in between these cells, really to eliminate any paracellular movement. And then on top of that, we've got the foot process of the astrocyte really securing that junction as well. So the foot processes of the astrocytes sit on top of the endothelial cell and really guard what's able to come out and enter the interstitial fluid around the brain. Okay, things like proteins are too large to slip through and enter the brain. This is why it's so much lower in the brain. Glucose is another example. Glucose has to go through by certain transporters in order to appropriately get into the interstitial fluid and be used as fuel in the brain. It cannot just go through the junctions, and it cannot pass through the astrocytes. We also have smaller hydrophobic solutes that are able to pass through the brain. Things like certain compounds, certain medications are going to be easier able to go through and be absorbed in the brain, whereas some of our more bulky compounds and molecules will not. Okay. The fourth learning objective is to identify the myelin sheath, talk about how it is formed in the central and peripheral nervous system, and just compare those two cells and those two processes. So let's start by talking about myelin formation in the central nervous system. Remember, this is simply brain and spinal cord. So myelin is produced by oligodendrocytes in the CNS. One oligodendrocyte is going to send out several extensions to different axons. So here is an example of that happening. Here is one oligodendrocyte. Notice it's sending out two different projections, but it can be more, it can be many more, right? We can have three as we see here. And it's going to just produce the myelin sheath. Now notice for the central nervous system, this is not going to be considered neurolemma. Neurolemma is exclusive to the peripheral nervous system, but I really want to make the distinction here that in the central nervous system, myelin is produced not with the inclusion of neurolemma. Okay, myelin is really the tissue that coats the axon. So as this oligodendrocyte releases these projections, they basically provide these layers of fat. That's all that myelin is, a fatty layer that's being produced around the axon at intervals. And it's going to appear white on imaging as well as on gross inspection of the brain. And so the myelin is going to be the axons. Okay, the gray matter is going to be the cell bodies, the dendrites that are going to lack myelin. Okay, so the myelin sheath, someone's asking what is neurolemma. I will talk about neurolemma in just a few minutes. It's coming up on the next slide. The reason I pointed out here is really to make the point that it's not seen with oligodendrocytes and the central nervous system. It will be seen in the peripheral nervous system with Schwann cells, and I'll kind of describe more further what it is there. But just make a note that it's not in the central nervous system. And then the last thing to say about gray matter versus white matter is just remembering that connection or that that contrast of the way that gray matter and white matter are organized differently in the spinal cord versus the way it's organized in the brain, right? Remember, in the brain, it's over top the white matter, the gray matter is over top the white matter. In the spinal cord, the gray matter is inside and the white matter is outside. But just to make the connection of what the difference is, gray matter is the somas and the cell bodies that are not myelinated. White matter are the axons, which are myelinated. Okay, now we get to neurolemma and myelin. Neurolemma is a structure that's required for the mechanism involving myelination specifically to the peripheral nervous system and the involvement of Schwann cells. So it's only seen in the peripheral nervous system and it's only made by Schwann cells. It is essentially a layer that's wrapping around the neuron that is being myelinated. Okay, so in order for myelin to be made in the peripheral nervous system, we have to have a Schwann cell. Here's what that cell looks like. Here's what that cell looks like. This is the cell nucleus of that Schwann cell. The neurolemma is the layer that projects around the Schwann cell itself. And it's really more specifically the projections that are coming out that actually wrap around the neuron. So this would be considered the neurolemma, the layer around the projections that are kind of engulfing or hugging around the neuron. And having the neurolemma wrap around the neuron is what is responsible for the production of myelin. So as we can see, this is how it starts out, right? And then eventually the myelin is laid down in these circular layers around the axon itself. So the blue structure here is the axon. This structure is the Schwann cell. This is the neurolemma, right? Only the outer layer is the neurolemma. And then the layers, the thicker layers that are being developed in between the neurolemma and the axon is what's considered the myelin. Okay, as far as the images down here, notice we've got the neurolemma here. In between the neurolemma and the actual axon, we have the myelin that's being produced by the neurolemma. But I really want to make the distinction that the neurolemma is not the myelin. They're different. The neurolemma forms the myelin, but the myelin, the neuron can be unmyelinated and still have a neurolemma around it. All right, so here is what an unmyelinated axon looks like. Notice it's a thinner layer versus a thicker layer here, thinner layer there. So notice we still have neurolemma, that layer around the projection that covers the axon, even though there's an absence of actual myelin. Okay, so here are some other examples of looking at unmyelinated axons. So here are some unmyelinated ones. Notice they still would have what's considered neurolemma around it. Here are some of the cell bodies or nuclei, excuse me, of the surrounding Schwann cells. Here's what a myelinated axon would look like, right? Here are the actual cytoplasm and nucleus of the Schwann cell that's wrapping around. And then here is the axon in question. And then this thick layer would be the actual myelin. Remember, the purpose of myelin is to conduct impulses more rapidly. In other words, to speed up, by way of insulation, speed up the saltatory conduction of action potentials along the length of the axon. In other words, speeding up the speed or the rate of the signal transduction.